For chapter 24, we're really going to focus mostly on metabolism and a little bit of energy balance, but certainly this first part 1 here, and chapter 24 will only have 2 parts, but part 1 will be focused more on the metabolism part. So as an overview for metabolism, I just wanted to remind you guys of the nutrients that we would be ingesting in our food. Of course, all the stuff we talked about in chapter 23 would be how we digest each of these and then absorb them. So we talked a little bit about digestion, not as much in the absorption part, but those are the nutrients that we would be getting out of our food. And of course, we would mostly be using these nutrients as some type of fuel for metabolism, which we're going to get into that more here in just a little bit. But I do want to say, again, I've been having trouble with my PowerPoint conversion anytime I record a slide and then convert it and upload it to YouTube. The second slide, for some reason, has not been showing up properly. I have no clue what the problem is. So and I don't see it until after I convert it. So right now I see my slide perfect, but you may on playback on YouTube be seeing not what I'm seeing. So if you're having trouble with just that second slide, if it may be correct, I don't know, but if it's not correct, just pull up the other PowerPoint just to look at what I'm looking at, because again, I just don't have time to troubleshoot because I'm trying to get you guys content as fast as possible, okay? So I do apologize, but we're gonna have to work around it. So what I do want to say about the primary nutrients, we've got carbohydrates, lipids, and proteins. All right, so carbohydrates mostly are going to be used as that fuel for metabolism. That's their primary job. Lipids we can use as fuel for metabolism, but we have lots of other jobs of lipids as well. Lipids are going to help us with our fat storage, which is good for insulation and protection. We use lipids to make certain steroid hormones like testosterone and estrogen. So lipids are used for multiple things. Proteins as well. That's sort of a last-ditch effort for metabolism to use as fuel, and we will talk about that. Proteins are usually just to make up things like muscles is what I meant to say. Proteins make up our muscles. Proteins make up, things like our hair and our skin, like the keratin in our skin, and proteins are used as enzymes in our body. Lots of different things for the use of proteins. Vitamins, we have a lot of different usage of various vitamins. Many of them are used for metabolism, but we have other vitamins like vitamin K is really useful for blood clotting. Vitamin A, you have to have it to have proper vision. Vitamin D is necessary for calcium absorption. So there's lots of different usages for vitamins, but we've got to bring them into the body. And we also have various minerals. Okay, minerals are going to be things like calcium and phosphorus, of course, necessary for many things: bone strength, synaptic transmission, muscle contraction. Those are the usages of calcium for sure. We've got things like iron. Iron's necessary component of your hemoglobin. Iodine, necessary for thyroid hormone. So all those different minerals. So we need to bring in all these nutrients in our diet, and then we use them for metabolism, which is what we're going to talk about on the next slide. So as a review, the term metabolism, a lot of times we think of it just as our ability to to maintain our weight. A lot of times, if you have a high metabolism, you can eat all the junk food you want and still not gain weight. Okay, a lot of times that's how we think of metabolism. I think of my metabolism, I may have told you guys this joke before, but I wish it was an actual joke. But when I think of my metabolism, I think of something that's like tanking in my old age, that kind of thing. But for scientific and physiological purposes, metabolism is more than that. Metabolism is going to be all of the chemical reactions that are happening inside of your body at any time. A lot of those chemical reactions are involved in energy storage, energy usage, okay, which is kind of getting down into the idea of what you eat, your diet, and then your weight. But it's everything, every reaction that's happening in your body goes to metabolism, okay? And metabolism can also be broken down into two different processes. You either have anabolism or catabolism, all right? Anabolism is the reactions that are used to build molecules. For example, if we were going to build a protein, we would take the building blocks of that protein, which are amino acids. We would take those building blocks and we would basically glue them together through a chemical reaction. And so we would then form a chain of 2 amino acids. Then we would glue the 3rd amino acid. And each of these are processes of anabolism. And so we're building bigger molecules from the building blocks. Okay, if it was a carbohydrate, we would be taking monosaccharides and glue those together to make a disaccharide, okay, or a polysaccharide if we had more of those saccharides put together. So anabolic reactions store energy in those bonds that you're making. Now on the flip side, you have catabolism. Catabolic reactions are taking these big molecules and chopping them up into their building blocks. So we could take a big long protein that has lots of amino acids and we could chop amino acids off of it, break the bonds that bind those together, and when you break those bonds, you release the energy that has been stored. All right, and so that would be a catabolic reaction that's going to release energy. Okay, so the way I think about this is anabolism makes me think of anabolic steroids. And when someone takes steroids, they get real big and bulky and they're building all this extra muscle. So an anabolic reaction is one that builds bigger molecules. That's the way I remember that. So when we talk about metabolism, we are going to, in this chapter, in terms of energy balance, we're going to talk about how we can use the different molecules in metabolism to get energy out of them. Okay, so the first molecule that we can use, and we do use very frequently as fuel for metabolism, is carbohydrates. The first part, the most common way that we use carbohydrates is through the process of cellular respiration. Now, you covered cellular respiration in chapter 3 in A&P 1. Now, if you remember, there was like a couple of pages, about 10 pages from chapter 24, that most of our A&P 1 teachers slide over into chapter 3. Okay, that may not be familiar to you. You may have forgotten it. Okay, it's a really difficult concept. And so what I have done is given you a supplemental video that has a review of cellular respiration, just so you can remember, oh yeah, cellular respiration, we're taking glucose and we're converting it into a lot of different things through glycolysis and the Krebs cycle and oxidative phosphorylation, and we're getting ATP out of it. okay? And we use that ATP for energy. So I've given you a separate video. If you want to stop right now and go to that video and read and listen to it, it may help you understand the rest of what we're going to cover in part 1, okay? Because I'm going to refer back to glycolysis and Krebs cycle a lot over the next couple of slides. So it's probably going to be beneficial to you to go back through and remind yourself about all of that good stuff, okay? All right. So we use carbohydrates to get ATP, all right? And that's through the process of cellular respiration. But we can do other things with those carbohydrates. And the thing that we're getting to here is that we've got to have glucose. And that glucose is going to be used for energy. So how, if we're not actively like eating food and getting the glucose that way, what are we doing with the glucose? Where does the glucose go? So we've got cellular respiration. That's immediate need, immediate usage of glucose for ATP production. But let's say we ate like a huge carb-rich meal. We used all the glucose we needed, but we still have some left over. Well, we don't want to just lose it. We want to be able to use it later. So what we will do is the process of glycogenesis. Okay, glycogenesis. Now, this is one of those times, this chapter is one of those times where you're going to get a lot of terms thrown at you. Memorizing them is definitely necessary, but instead of just memorizing the words, because they sound a lot alike, break down the words to what they mean, like the pieces of the words. All right, and there's some work on your mastering that I have you do that a little bit because it helps a lot. All right, so for glycogenesis, this is when you have excess glucose, but you don't need all that glucose right now in your body to be used, but you don't want to get rid of it. So we store it, okay? And the form that we store glucose in our body is in the form of glycogen. Glycogen is just a whole bunch of chains of glucose. Literally, they're all holding hands, all right? So we convert the glucose and basically just add them all together, glue them all together, and we store it in the form of glycogen. So glycogenesis, if you think of those two separate words, glyco stands for glycogen because that's the first part of that word, glyco, and genesis is the production. So glycogenesis is the production of glycogen. All right, and so from there you just need to understand why we would make glycogen, storage for glucose. We mostly store glycogen in our liver and our skeletal muscle, and we're storing it for later. We need energy later when we don't actively have glucose coming into our body, and we'll talk a little bit more about that in part 2. So like what happens when we eat versus when we're in like a starvation, not eating mode. So we'll talk about how all this works together later. Okay. So the next thing that can happen is glycogenolysis, okay? You could think of that, I like to think of it, and I will say it in that way, glycogenolysis. But it can also be said as glycogenolysis, all right, glycogenolysis. I think of it as glycogen lysis because that's exactly what it is. Lysis is a term that means to break. And of course, glycogen in this case, you've got the whole word there, it's glycogen. We're breaking glycogen. So why do you need to break glycogen? This would be when you are needing more energy, you need more glucose, but you're not actively eating it. So you've got to go into your stores of it. So you're breaking into your glycogen stores by breaking the glycogen and releasing the glucose. Now, the liver glycogen storage can be used systemically for all of your body cells when you need that glucose, but again, you're not actively ingesting it. Any storage of glycogen in the skeletal muscle, when it gets broken into, it really is just used within the skeletal muscle. All right, so that would be when you're exercising or, you're using your muscles for whatever reason, in a fight or flight type of situation, and you need more energy specifically within the muscles. And so the storage of glycogen in the skeletal muscle, when it is broken into glycogenolysis, then it just stays in the skeletal muscle, that glucose. The third thing that we can do to metabolize carbohydrates is called gluconeogenesis. Okay, gluconeogenesis. What this is when you make glucose from a non-carbohydrate substrate. And we would call that de novo. That means like from the beginning or anew. So in this case, we need glucose. And we'll talk a little bit more about why we need glucose specifically. But remember, glucose is giving us that fuel for cellular respiration. It's giving us the ability to make ATP. And some cells almost extensively rely on glucose in order to get ATP. So you've got to have glucose all the time. But if you've run out of glucose or run out of glycogen, then you've got to get glucose from somewhere. So you can make glucose from molecules that are not carbohydrates, and that would be gluconeogenesis. So again, we're talking about glucogenesis. So genesis is the production. Gluco is referring to glucose specifically. And then the neo part, that's the new production part. And a lot of times, gluconeogenesis is making glucose from proteins, which is kind of a weird thing, to make glucose from proteins. So we're going to talk about that process in a couple of slides. And that is primarily achieved through the liver. We can also use lipids for metabolism, so to generate ATP molecules, but we don't always use that. We don't always use those lipids. A lot of times we rely on the carbohydrates, but we can use the lipids. We, as I said earlier in this PowerPoint, we use lipids for a lot of different things. So we use them to make our steroid hormones, phospholipids, which are part of the plasma membrane. We need a layer of lipids for storage, for insulation, and for to keep our body temperature and to protect a lot of our organs. So a lot of times we use lipids for those reasons, but we can use them for metabolism to get ATP. And what they say is that if you are exercising at about that 30-minute mark is where you would switch into using lipids as a source for your metabolism. So you would burn off lipids at that point. Usually after your glucose and your glycogen stores have been used up. But in all actuality, lipids will provide us with a little over 2 times of the energy when you directly compare it to a carbohydrate. So we can use it for sure. We just don't use it a lot. But lipids in our bodies are mostly stored as triglycerides, okay? If that word and a lot of these other words like glucose and saccharide and amino acid, if those don't sound familiar to you, please go back to chapter 2 and just read it again. Review all those words so you know what I'm talking about, okay? It's really important. Nonetheless, triglycerides, when they're are broken down into a molecule of glycerol, which is a 3-carbon structure, and fatty acids. Fatty acids are these long chains of carbons with some other stuff, some hydrogens, and we've got some oxygens thrown in there too. But what we do is, in terms of metabolism, so trying to get energy out of these molecules, we can take glycerol and we can convert it to an intermediate product in the process of glycolysis. Remember, glycolysis is a process of like 6 steps, and it's the first part of cellular respiration, and we're trying to get glucose to become pyruvic acid. And so one of the intermediate products is called glyceraldehyde 3-phosphate. And so we can convert glycerol into that molecule, which can then be used to pick up glycolysis wherever it enters into the process. And it will continue through the process of generating ATP. It may not generate the same amount of ATP as a glucose molecule would, but it can help. The other thing that happens is those fatty acids, a triglyceride has 3 fatty acids on it. We can convert the fatty acids to cellular respiration intermediates through the process of beta oxidation. So beta oxidation is a process that happens in the mitochondria, and it takes those fatty acids. Just for an example, let's just say you have a chain of 20 carbons that is a fatty acid. Okay, long chain, 20 carbons. What beta oxidation does is it's going to break the end 2 carbons off of that 20 carbon fatty acid. So what you're left with is an 18 carbon fatty acid and a molecule that is converted into acetyl-CoA. Okay, and acetyl-CoA is a 2-carbon structure. Okay, it's got a couple of things added to it. We'll see it on the next slide, but it's a 2-carbon structure. And if you remember, acetyl-CoA is the molecule that enters in and begins the Krebs cycle or the citric acid cycle. Those 2 names mean the same. Okay, so let's look at that on the next slide, beta oxidation. Here we can see beta oxidation as well as taking that glycerol and converting it. So we have our lipids, which are mostly triglycerides. On the left, you can follow what happens to glycerol, which what I mentioned on the previous slide there is it's converted to an intermediate, which is glyceraldehyde 3-phosphate or G3P. And then through the remainder of the process of glycolysis is converted into pyruvic acid, which then as pyruvic acid moves into the mitochondria, it creates acetyl-CoA. Okay, if none of that sounds familiar, go back, review cellular respiration. All right, so the fatty acids, go back to the top of the slide on the right. The fatty acids go through the process of beta oxidation. All right, this beta oxidation, like I said, let's just say we have a 20 carbon molecule, and the process of beta oxidation chops off the end 2 molecules. All right, so you've got a 2 carbon molecule, and through adding all of those different things, so you have to use ATP to make this happen. Okay, you have to use ATP, but you also add what we see, that coenzyme A, you see some NADH and some FADH2 being created. Those are important molecules for oxidative phosphorylation in terms of energy and ATP generation. But what happens is that 2 carbon molecule gets converted into a molecule of acetyl-CoA. Okay, the same thing that we just talked about, it will then continue through cellular respiration. But what happens with beta oxidation is if you have any fatty acid left, the process of beta oxidation happens again. So it'll go back to that 18 carbon molecule, that 18 carbon fatty acid, chop the next 2 carbons off of it. So you have a 16 carbon molecule and another 2 carbon molecule that will go through the same steps you see on the slide and form another molecule of acetyl-CoA. and so on and so forth. So a 20 molecule, 20 carbon fatty acid would turn into 10 molecules of acetyl-CoA. The whole time using ATP, yes, but also creating more NADH and more FADH2, which can go to oxidative phosphorylation and make more ATP. And those acetyl-CoAs can make and will go on through Krebs cycle and make a good bit of ATP as well. Okay, so even though you're using ATP in this process, you are generating a lot of ATP, more so than a little glucose might. But remember, we need lipids for other reasons, so we don't always use them for metabolism. We can do other things with those lipids. If we ingest a big meal of lipids and we don't need them all, then what we're going to do is undergo lipogenesis. Okay, lipo is referring to lipids, genesis is generation. And what this is you've got all the glucose that you need, so that's a part of this too. You don't need any more ATP, but you have all this extra lipid molecule. So you're going to convert it into a storage in order to use later. Okay, so lipogenesis is going to be taking those extra fats, extra lipids, converting them into a storage form. And that's going to occur a lot in our adipose tissue because that's mostly where we store our lipids, but this can also happen in the liver as well. So we're using, if we think about us using glucose and we use all the carbohydrates that we have, but we're not actively eating, so this would be sort of in between meals. We've used all of our carbohydrates, but we still need ATP. That's when we would want to then break into our fat storage. And breaking into our fat storage is called lipolysis. Also, you can think of it as lipolysis, because that's the two roots of the word. Lysis is the breakdown or the release, and of course the lipo is referring to the lipid storage. All right. So that's when we would work to use our fat storage. Again, the carbohydrate issue is sort of a separate one, but in this case, we would have used all of our glucose, used all of our glycogen, but we still have energy demands. Okay, so like for example, I mentioned after about 30 minutes of exercise, you got to break into your fat storage. This is the process of doing that, lipolysis, all right? But we could have something that happens with lipolysis that might not be such a good thing. What I mean by that is that the process of ketogenesis can occur. And if you're thinking about lipids being metabolized through beta oxidation, you are making acetyl-CoA, all right? And then acetyl-CoA would normally go on through the Krebs cycle and work to generate ATP. But if you have an issue in which you don't have enough carbohydrates in order to make up the other parts of Krebs cycle, then you can't use that acetyl-CoA. And so what happens is that you go into something called ketogenesis or ketosis. So that's what we're going to talk about on the next slide. When this gets to be a problem, it's usually in people that have diabetes. And so it can cause a condition called diabetic ketosis or ketoacidosis or metabolic acidosis. All those things really are referring back to the same this same issue. So remember, the issue with people that are diabetic is that they are having trouble with producing insulin, or either sensitivity of insulin. Either way, they don't have insulin and they are not able to take the glucose in their blood and move it into their tissues to be used for ATP generation. And so what can happen if you have like a lot of carbohydrates in the diet, or if someone's not taking their insulin correctly, or a combination of those two things, you're going to go into where you would want to break your fat storage down. And if you remember, that's the process of lipolysis. And at the same time, you also, your body thinks that it needs glucose because it's just not getting it in the cells. And so you actually break down your glycogen storages as well, so glycogenolysis. But those two things are happening in the background. And so what happens is that those people would start to break down their fat, and so the process of beta oxidation would really ramp up. And remember, the end product of beta oxidation is acetyl-CoA, because again, your body needs energy and it thinks it can't get it because it really physically can't. You've got all this glucose, you just can't get it into the cells, which is the job of insulin. So you start to use acetyl-CoA, but you're making it too quickly. And so you've got this acetyl-CoA that is building up because you just can't use it as fast as you're making it. Also, you're having trouble making the intermediates of the Krebs cycle as well, because they're all different carbohydrates. You don't have glucose in your cells, so it's sort of a perfect storm of all of this. And so what happens is that the acetyl-CoA gets converted into something called ketone bodies. Ketone bodies are very acidic. And so what this does, the ketone bodies, is it causes your blood pH to decrease, become more acidic. And the other thing that happens is that those ketone bodies, and also all the extra glucose that stayed in your blood, it starts to collect in your urine, okay? So you've got ketone bodies and glucose in your blood, okay? Those ketone bodies are making it more acidic, and you've got ketone bodies and glucose building up in your urine. So your blood pH drops, like I just said, and one of the signature symptoms of ketoacidosis is a fruity breath smell, and that's from the ketone bodies. Okay, for whatever reason, they have sort of a fruity smell to them. So if a person that's in diabetic ketoacidosis is close enough to you and you smell their breath and it's fruity, you know for sure that that's what's going on. But all of these things that are happening in the body lead to these other symptoms. You start urinating more because of all that glucose and all that ketone bodies, those ketone bodies in your urine, which dehydrates you severely, making you more thirsty to try to offset that dehydration. It makes you breathe very fast, and that's in efforts to try to neutralize your blood pH again. Think back to our discussions back in the respiratory system about respiratory acidosis and what kinds of things happen to try to fix that. So it's kind of the same thing. And ultimately, if this is not dealt with by basically getting insulin in and neutralizing that blood glucose, this could lead to some really, really serious things. So diabetic ketoacidosis is really no joke. It's nothing to mess around with. And it's a process of things that's happening, but it's all sort of boiling down to acetyl-CoA and this idea of increased usage of beta oxidation. All right, guys, here is the slide. This slide has so much going on. It is not even funny. It will literally blow your mind out of the water. Everything that we have just talked about in terms of using glucose for cellular respiration, triglycerides for cellular respiration, storing these molecules, forming ketone bodies, everything that you could think of is on this slide. There's lots of terms like lipolysis and lipogenesis. There is beta oxidation. All of it is here with your in terms of like lipids and how it moves in excuse me, to cellular respiration. So please take a number of minutes and walk yourself through this slide and make sure you understand the terms and how all of those terms fit in with metabolism. Remember, metabolism is taking the chemical reactions and converting things and using those things for energy and ATP. Okay, so this slide is certainly important for you to look through. Make sure that you understand, but I'm going to assume you can do that on your own time. Go back through the previous number of slides. If you see lipolysis, go back through and read about lipolysis and make sure that you understand what is happening and why that might happen. Okay, same thing with beta oxidation and so on and so forth. The last nutrient we need to talk about in terms of metabolism is proteins. So whereas extra carbohydrates and lipids can be stored, excess proteins cannot be restored. All right, so if we have extra proteins, we can use the amino acids. There are lots of different proteins in which they have to replace their amino acids. We're always making new enzymes. We're always repairing our muscles, so that's adding protein. So we can reuse the amino acids, okay? And we also could potentially use these proteins to make ATP. That is a last-ditch effort, okay? We don't frequently use proteins to make ATP unless we are in dire need of energy, okay? We are so out of glucose, we're so out of fat that we start breaking down proteins in order to generate ATP. Okay, that would be like a serious fasting situation, and we would start breaking down like muscle to get the proteins out of there and make ATP. So let's look at the process that would occur to have this happen. And it's a two-step process, and it's the process of transamination and deamination. All right, all this is happening in the liver, and just, hang with me because this one can get a little bit confusing, okay? So, transamination is first. So what we're going to do is we are going to use something called a keto acid, all right? This is not a ketone body, something totally different, so don't get those two things confused. But this is a keto acid. And that keto acid, we could use it to move into cellular respiration at various stages and generate ATP. That is something that we can do if we are in need. All right, but we'll also see another way we can use this keto acid. But the process of transamination, I'm going to talk you through specifics on the next slide, by the way. But the keto acid is what is produced in the transamination process, okay? And that keto acid could be used for ATP production. But the other thing that is produced in transamination is something called glutamic acid, okay? Glutamic acid continues through stage 2, which is the process of deamination. All right. Deamination is when we're trying to remove the amine group. Okay. Transamination, that first step, trans is going to kind of refer to like a change. So in this case, the amine group is just changing from one molecule to the next. But in this case, we want to deaminate. We want to remove the amine and get rid of it. All right. That's sort of the whole reason we're doing this process as sort of like a background, something that's always happening. If we have too many proteins, that means we've got too many amino acids. Amino acids have a molecule of nitrogen in them, sometimes more than one. And this, our nitrogenous waste, this is the way we get rid of nitrogenous waste, is through transamination followed by deamination. So in this case, the amine group is going to be removed and it forms ammonia, which we're going to see on the next slide. And that ammonia is converted into a molecule called urea. And urea is what is a primary component of urine. That's what we pee out. All right. But in the deamination process, the other thing that's left over, the keto acid, as we talked about on the previous slide, it could go straight into cellular respiration, or the keto acid could restart this whole process of transamination and deamination. Okay, so there's sort of two different scenarios that can happen here. So let's look at some more specifics on the next slide and talk through it one more time. Keep in mind when this is happening, this whole trans versus deamination, we've got too many amino acids. Okay, we can't store them, so we got to get rid of them somehow. All right, so in this case, the first case scenario we're going to talk about here is just your typical, let's get rid of an amino acid because we don't need it, maybe it's damaged, let's just get rid of it. The problem is it has nitrogen in it. Okay, nitrogen has to be dealt with in a very specific way. So here's what we do. The first thing is transamination. The amine group of the amino acid is what has nitrogen in it. All right, so if you look at the very top, you see transamination, you see this long equation. The amino acid is where the amine group is, that nitrogen. Okay, so we are going to take the amine group off of the amino acid and we're going to switch it to a keto acid. Okay, so we have two substrates, the amino acid and the keto acid. And when we switch the amine group, we make two products. The first product is a different keto acid, and that's the confusing part here, okay? We start with a keto acid and we make a keto acid because there's multiple different forms of keto acids, and we're just not going to get into all the specifics. So we're going to come back to that keto acid here in just a minute, but the other thing that we make is glutamic acid. Okay, glutamic acid is one of our 20 amino acids. Glutamic acid continues through step 2, the process of deamination. We take the glutamic acid and we are going to remove a part of it to make ammonia. And then that ammonia, which is going to have our amine group, that NH3, we add CO2 to that. And that's how we make urea. Urea then goes to the kidney and we pee it out. So that's how we would get rid of extra nitrogen, get rid of extra amino acids. If that's the only thing that is the problem, then if you go way back up to the top, the transamination, the products, keto acid and glutamic acid. We talked about what happens to the glutamic acid. If we're just trying to get rid of excess proteins, the keto acid goes back and goes through the whole process of transamination again. All right, as long as there's excess amino acids, that can continue to happen. But here is a second case scenario. The second case scenario is that we're in a state of fasting or a state of extreme starvation. In this case, we need glucose. And we need ATP and we need a way to make ATP. And so what we're going to do now is take that keto acid that was made through transamination. If you follow that wavy line over to the left of our picture of the liver, we can take those keto acids, modify them slightly, and they become intermediates of the Krebs cycle. And what that means is they enter into the Krebs cycle at various stages, but they continue through the remainder of cellular respiration. So they're going to make more ATP for us. So in this way, we would be taking proteins that we've broken down into their individual amino acids. We've transaminated them to form a keto acid, and then we take that keto acid and use it to make ATP. Again, the reason you would want to do that is because you're lacking on glucose, probably lacking on fats as well. Again, this is a last-ditch effort, extreme starvation. All right, so this is where we're going to stop for part 1. I'm going to pick back up with part 2, and we're going to talk about those different states of, how we get energy and what molecules we use when we're eating versus in sort of a fasting, non-eating stage.